So thanks again, everyone, for joining to today's seminar on Reimagine How You Study the Brain. My name is Yvonne Peterman. I'm a science and technology advisor with 10x Genomics, and we are running this seminar today with Millennium Science, our distributor here in Australia. And I'm going to use the first about 40 minutes for the presentation to introduce the technology to you with specific neuroscience focused examples. And then I'll hand it over to Marta, who is going to explain to you in much more detail how our new Millennium Science 10x Genomics Fellowship Program is going to work, how you can access it, how you can apply, etc. But with this seminar, I just want to give you some ideas and spark your imagination on what the 10x Genomics technology can actually do for you in the neuroscience field. And to kick it off, we just going into the fact that the scientific and medical community still, despite a lot of research that's already been conducted, is still only understanding a very small fraction of the actual full complexity of biology and the complexity of the brain specifically. As we know, the human brain is very complex. It consists of over 170 billion cells, each with a genome of 3 billion DNA base pairs and a unique epigenetic program regulating the transcription of tens of thousands of different RNAs, which are then translated into tens of thousands of different proteins. And really understanding these complexities at high resolution is the key to genomic discoveries and ultimately accelerating the disease and therapeutic studies that are occurring. And the 10x we've set out to really drive this acceleration through expanding our single cell and spatial application portfolio, because we want to enable you to dive into this portfolio with your data, with your samples, and really drive and generate data. And with this fellowship that we are opening up now, we really hope that we can actually make this step getting into it even easier, to help you with design of your experiment and really guide you through the entire process from sample to data. We have seen a number of breakthrough discoveries across several different research areas in neuroscience research over the past years, where the 10x technologies were instrumental. So you can really be, driving off and feeding off the information and the expertise that we have already got out there in this field. And researchers have helped, to get a lot closer to an understanding of complexities behind specific diseases, such as schizophrenia or Alzheimer's disease. And our solutions have helped uncover some of the most fundamental processes that occur in human biology, and including the early stages of development, mechanistic insights into normal brain function, and key pathways that are involved in neuropathologies. So behind discoveries that we are reading about are these complementary 10x Genomics platforms that are listed here. And currently we have two established technology platforms in the market, the Chromium single cell and the Visium Spatial. Both are providing you with next generation sequencing based readout. And we are constantly bringing new innovations in the form of new improved kits and instruments to these portfolios to make samples more accessible and analysis even more affordable. And for the scope and purpose of today's seminar, I'm going to focus on these two going forward, as these will be accessible to you through the fellowship program. But I do just want to mention our third platform that will be arriving in the market early in January next year, which is our Xenium in situ platform. It will allow you to look at RNA expression in tissue sections at subcellular level This is going to be microscopy based, and it will really allow you to go from discovery, which is what the Chromium single cell and the Visium spatial solutions enable you, more into the focused deep tissue analysis of very specific genes using panels. So focusing on the first two now, our assays can really be broadly associated with these two, beginning with the Chromium single cell platform. Now, as you can see here, it consists of an instrument that is either the Chromium controller, the Chromium iX, or the Chromium X instrument. And it's quite small. They're small benchtop instruments that really integrate easily into your lab. And we have a number of service providers out there in Australia also that can, handle samples and process single cell samples for you. So into this instrument, you feed the disposable microfluidic chips that you see here on the left, and then on these chips you can pick, one of 5, or you can pick 5 main assay types that we have, and I go into a bit more detail later on. But irrespective of what assay you actually pick for this instrument, the key output from this platform is the ability to characterize cellular heterogeneity of your sample. And your sample is, what you input into this instrument, that it can either be a single cell suspension or a single nuclei suspension. And really, how the quality of the suspension is really defining the quality of your experiment. So we spend a lot of time, talking to you about how to generate that single cell suspension, how to collect your sample, how to store the sample, and how to process it accordingly. So there's a lot of thought that goes into this part of the workflow. Now, the second major technology platform is our Visium spatial transcriptomic platform. And unlike the Chromium platform, this technology now uses whole tissue section. So you're not dissociating your tissue anymore. You're taking the section, and it can be fresh frozen or FFPE section, and you're analyzing this now on a specific spatial transcriptomic slide. That one of the examples you can see here in the picture. Currently, you do not need an external instrument to perform this workflow. It is manual, and we have recently made an addition to this workflow with our CytAssist instrument, which will enhance the ability of utilizing a much broader range of tissue types or tissue or sample types for you, and I also cover that a bit later in the presentation today, what that really means. So what kind of fundamental questions can be answered applying these two technologies, specifically in neuroscience? And here we have listed a few of these that keep coming up and that are being addressed specifically in the neurodegenerative disease research area. So there's often the question, does the protein aggregation begin within a select or a finite group of cells? What's the relationship between disease onset and progression? What underlies the phenotypic diversity of pathological proteins, etc.? So there is a lot of sort of key questions that are still being asked, and where the technology can help. And here we've tried to put together specifically showcasing how the portfolio that we have for multiomic analysis, meaning analysis on top of just studying single cell gene expression, how can this answer these particular questions that I've just showcased? And you can approach this with all the different assays that we have. You can look at RNA plus chromatin with our multiome kit. So we are looking here at the accessible chromatin regions, for example, in the nuclei. And you can, with this technology, you can identify disease associated with genes and explore gene regulation in general. With just standard single cell gene expression, you have the ability to characterize cellular heterogeneity in your sample, as I mentioned, and you can also identify novel cell types and states in your sample. With our single-cell immune profiling solution, you really can dive into the T and B cell receptor composition of the sample, and that takes you to biomarker discovery as well as explore disease pathogenesis specifically from the immune profiling point of view. We can also analyze protein expression using, for example, the cell surface protein detection that we've got inbuilt with our feature barcoding technology, looking at, or helping with cell state deconvolution and analyzing transcriptome proteome interactions. So we've seen some of these toolkits especially applied, and some of these cases, some of these questions have been answered. And we've just put here some examples of these publications that address these kinds of research questions, looking at the combination of kits on the Chromium instruments. And to go into one of these in a little bit more detail is this study here published in Nature Genetics, using or doing the single cell nuclei sequencing as well as ATAC-seq analysis. And what this is showing here is the first study that's integrating single cell gene expression and single cell ATAC in human Alzheimer's disease, in human Alzheimer's disease brains. And by combining the gene expression and the chromatin accessibility data here, we actually get the idea or the capability of identifying actionable therapeutic targets. And how this was conducted, the researchers analyzed brain, utilized nuclei, and integrated these two data sets, nuclei and single-cell ATAC-seq. So that was done before our multiome kit was available. And they could identify gene regulatory landscapes. They were able to see or highlight the role of glia cells in Alzheimer's disease pathophysiology, and identified several genes that will be further studied in the pathogenesis. On the same note of Alzheimer's disease researchers, I wanted to point you to this resource here, which is an application note that we have published, studying single-cell and spatial multiomics in Alzheimer's disease and identifying specific disease markers by combining these two assays. At the bottom here, you can see the type of tissue that we've used and the assays we've used and what we've studied by doing the multiomic integration. And just here is a quick highlight on the actual study design. So we've used an Alzheimer's disease mouse model to study. We've analyzed 12 mouse brains that were collected with different time points, as you can see here, spanning early, middle, and late life. We use 2 replicates per time point from these mouse models, and then analyze these either with a single cell ATAC or with the spatial gene expression solution. And some of the highlights are listed here. I've put the barcode there as well to take you to the actual application note for further reading, because it does go into a lot more detail around sample preparation, analysis, and handling, etc. So we can see here that the multiomic data analysis yielded new insights into the single cell chromatin and spatial gene expression differences throughout the time course here of the animals that we've chosen. And several Alzheimer's disease related markers were identified, and they showed differential expression between the transgenic and the wild type mice and microglia and oligodendrocytes at these different time points. And we've done motif enrichment analysis of these differentially accessible chromatin regions that were identified, or identified transcription factors involved with the amyloid plaque deposition. So this is quite an elaborate application note. We also go into detail on how the motif enrichment analysis was conducted. So this is a really good read as well when it comes to methodology. Just wanted to bring up this picture here that talks about or showcases some of the Visium spatial gene expression data, specifically in the discussing the plaque burden or where the plaques occur. We used immune fluorescence assessment of these regional plaque burden areas. Blue here is representing DAPI stain, and orange is our human amyloid precursor protein staining. And in B, this is something here where you can see close up a region on the Visium slide with our barcoded spots in the thalamus of 13 months old mouse with plaque in orange, and we are grouping here Visium spots based on the proximity to the actual plaque. And then we are looking here at some of the violin plots and a heat map, really showcasing the differentially expressed genes across these 3 plaque proximity regions. Another example of utilizing, two different assays on the Chromium controller instrument is this publication here, where we've used, where the authors have used single-cell gene expression and the single-cell T-cell receptor sequencing component to analyze, to also analyze Alzheimer's disease. So here they combined these 2 assays and investigated the role of the adaptive immune cells in Alzheimer's disease pathogenesis. And this study here, published in Science in 2019 by Vermashev et al., also looks at a neurodegenerative disease, or specifically autism in this case, and they looked at, or they had bulk gene expression data available, and they wanted to look specifically more at the genetic heterogeneity of autism by utilizing the single cell, deep dive into the samples. So they wanted to understand the underlying cell type specific molecular pathology of autism spectrum disorder in more detail, and utilized nuclei of 41 post-mortem tissue samples that they had access to and analyzed them as well as controls. And here, what they've been able to show in the graph C here, we can see unbiased clustering of these nuclear profiles from these ASD and control samples. And we can see that they've identified 17 unique cell types within the sample. And one notable difference was observed between the autism cases and the controls, and that was where relatively more protoplasmic astrocytes were present in ASD patients. Now, to switch from some of these examples that we are seeing more to going into our solution. What does it mean? What are our assays? What's the software, and how does the whole workflow work? And I just want to spend sort of the next few minutes going in a little bit more detail into all the parts of the workflow. And to start with, I actually want to highlight the fact that we have complete analysis and visualization support for every assay. We do see specifically when researchers get started with single cell and spatial analysis, the data analysis side is often providing the biggest hurdle and the biggest bottleneck when doing these kinds of experiments. And we have Cell Ranger and Space Ranger available to specifically analyze your data with preloaded pipelines. But we also have Loop Browser available, which is your interaction platform or visualization platform with your data. Both these are downloadable for free. Loop Browser actually runs on your laptop, so that's very easy to install, and you can actually download quite a large number of data sets that we have available. There is also a few neuroscience data sets that I can point you to, and you can open up these data sets and play with the data and see what the data reveals, and also what our Loop Browser software lets you analyze. And another word on the data is also that you can run it in Loop Browser, but the data format is universal, so you can take the data sets as well and analyze them with any third-party tool that's out there. And we know there's a plus 1,000 different types of analysis softwares out there actually that you can utilize for single cell data. So as I mentioned earlier, you can load one of the 5 assays that are listed here onto these disposable microfluidic chips. And I just want to say what sort of at high level, what the differences are between the 2 different kits. And with the first 2 assays, single cell and immune profiling assay, you can choose to measure gene expression plus a range of additional analytes. For example, cell surface proteins. This is conducted via antibodies that have a oligo hybridized to them. So we can read that barcode out in the single cell data set. And you can also measure single guide RNAs with CRISPR perturbation screening. We have that enabled now for the 3' gene expression as well as the 5' immune profiling solution. With the 5' immune profiling solution, you can dive deeper into the T and B cell receptors. You can really understand the clonality, and you can also do antigen detection as utilizing additional add-on modules for these assays. The single cell attack and multiome kits, they are our epigenetic solution. Here you are measuring the accessible chromatin in nuclei. In case of the multiome, you do add on that RNA gene expression from the exact same nuclei as well. And the last, our newest single cell kit that's listed here, it's our single cell gene expression flex kit. This is really a kit that we are really excited about. It's revolutionizing single cell gene expression analysis, and it's quickly becoming the new gold standard and brings you great benefits. One of them is the flexibility, and what we mean by that is it allows you to collect your sample and fix it at time of collection. So you're locking in your biological state, and then you can store your sample until you're ready and have collected enough samples to conduct your run. So that is flexibility that also allows you to keep really the biology of your sample intact. And another benefit you get is the high sensitivity, because this assay also works with the probe pool. And we have inbuilt barcoding and multiplexing capability, really allowing you to run your experiment a lot more cost effectively, but while also saving on sequencing cost. So that's really all I'm going to say about the single cell fixed RNA profiling kit at high level. The fixation can be done with whole tissue or cell suspensions or tissue culture, etc. It's a 4% formaldehyde solution that you're using following our specific protocol. So please let us know if you want to do this, exactly what sample types you're dealing with, and we can guide you specifically on how to preserve these cells. The multiplexing capability that I mentioned that we have built in allows you to scale up. You can actually run up to 16 samples per channel. However, I just want to say here, for the purpose of the fellowship, we do really encourage everyone to go and start small, start with a few samples, probably 4, we find as an initial experiment is a really good number to just really make sure you familiarize yourself with the technology and also with the kind of data that you're getting from it. And these higher multiplexing stages, that is sort of a tool that we recommend sort of for the larger follow-up studies. So it's highly sensitive gene expression measurement, as we said, and the sequencing cost that's required is actually halved with this kit. So just running into the core technology for molecular barcoding and partitioning, which is at the core of our technology, you would have probably seen these images many times, where your experiment starts with the dissociated cells or nuclei that you bring in, and you capture them in an emulsion with gel beads that we call gel bead in emulsion. And the cells are then lysed and RNA is reverse transcribed inside the gems. And during the steps, the cell content is barcoded by previously mentioned by these 10x barcodes that are in the gel beads. And in addition, every molecule is barcoded by the UMI that is present on the oligos on each of the beads. And at this point, you can then pool, generate sequencing libraries, and proceed with sequencing and data analysis. And really what makes this so exciting is, and what we're indicating here at the bottom of it, is that you can actually add on several modalities and analyze that at the same time. So for example, a cell surface protein, barcoded antibody, or a barcoded antigen. So the overall workflow for after the library generation just basically continues with the sequencing capability, or the sequencing, which is usually done on Illumina, NextSeq, or HiSeq instruments, and then you take the data and analyze them with either Cell Ranger in the single cell case or Space Ranger in Spatial transcriptomics and visualize your data on Loop Browser. And here at the bottom, we have a short video on how this partitioning occurs on the microfluidics. So now, with the introduction of the single cell technology, I want to switch gear and introduce the Visium spatial transcriptomic technology, first with an overall introduction, and then mentioning specifically our CytAssist instrument, which is our newest addition to this workflow, and really what it does. So Visium spatial gene expression can be performed on fresh frozen tissues, which is species agnostic, or FFPE mouse or human tissues. We are measuring gene expression by capturing polyadenylated RNA molecules directly in the case of the fresh frozen tissues, or via the whole transcriptome covering probe pools that are specifically designed to protein coding genes for human and mouse. So at the moment, both of these workflows, or up until very recently, both of these workflows are manual. And for the FFPE tissue handling component, we now have the CytAssist introduced into the workflow. But before I go into this, I want to mention that in addition to capturing the whole transcriptome, we also have the protein detection paired with gene expression enabled on Visium. We do this at the moment with immunofluorescence of up to 5 antibodies in the workflow, and you can measure and visualize gene expression plus immunofluorescence at the same time. In the future, the protein detection will be enabled via barcoded antibodies, and this will enable larger pools and sort of change into the sequencing readout for you that you'll get from the Visium dataset. So here we have an example of the brain using a NUN marker for NUN fluorescence marker and the specific RBFOX3 gene expression. You can see with Visium, you can overlay or you can visualize each individually, or overlay both protein and gene expression in the same image. With Visium spatial gene expression, up until about a year ago, we only had fresh frozen samples enabled, and we heard very loud and clear in the market that Visium needs to be capable of handling FFPE tissues, which we have enabled on the Visium spatial gene expression platform now in the manual way. And what we've also heard is that the way you handle FFPE tissues on our Visium gene expression slides has meant in a lot of cases no already stained and archived FFPE tissues were able to be used in this technology. And this is where our Visium CytAssist platform comes in. It lets you now actually use already pre-cut slides, and I might switch to the next slide because it highlights this a little bit. It really helps to get more out of an FFPE sample that you might already have. So we can now not only use a block and section a tissue onto a plain glass slide where you don't need to take care of placing the section into a very specific capture area. The section can just go on a plain glass slide and can then be applied to the CytAssist instrument. But you can also use tissue sections that have already been stained and cover slipped and archived and have been stored for a long time. You can recover these slides and use them for spatial gene expression analysis on the CytAssist instrument. What this also allows you to do is, use H&E or immunofluorescence staining and actually look at your tissue and look at specifically the area that you think is biologically the most interesting and the most useful to pick to study spatially. So once you've identified this, you can then use the section and apply this on the CytAssist, picking very specifically that area that you've identified. Due to the fact that you can align manually the slide with the capture area on the actual Visium slide. Because the key with this instrument is, I don't actually have a view in the slide deck here of the open instrument, but it opens up and you load up to 2 slides, up to 2 sections on a plain glass slide up the top, and at the bottom you load a very specific CytAssist spatial gene expression slide, and then you align your sample to the outline on the Visium spatial gene expression slide. And we have made another improvement in terms of the size of the capture area and in terms of the size of the tissue that you're now allowed to or able to use. We've increased this from 6.5 by 6.5 millimeters, which is here outlined in the little blue box, to 11 by 11 millimeters, which is outlined here in the dark gray box. So we've basically increased the area that you can analyze almost by threefold, really letting you get even more out of your sample. So CytAssist really helps with a lot of these things when you deal with FFPE samples. If you are analyzing fresh frozen samples, you perform the analysis still manually. However, going forward, the CytAssist will also be compatible with our fresh frozen tissue sections. And specifically for neuroscience, I want to mention we have seen in the field with researchers studying the brain or spinal cord that there have been detachment issues noticed on our Visium slides in the manual workflow, and this is actually something that by utilizing the CytAssist is now completely avoidable, simply because you are utilizing a standard glass slide with your section on it. You're hybridizing the probe pool to that slide, and inside the instrument, all you do is you actually transfer the analytes onto the CytAssist slide, onto the Visium spatial gene expression slide in the CytAssist. So there's no direct attaching to anything and really reducing the chance or the risk of any detachment issues. So really, CytAssist is quite useful for neuroscience samples. The structure of our Visium spatial gene expression slide is highlighted here on one of our manual slides that are still out there. And we can see here the 6.5 by 6.5 millimeter capture area. It holds about just under 5,000 barcoded spots. It's surrounded by a fiducial frame. This fiducial frame is really quite important when matching the histology image with the sequencing output image to map back the location or the origin of the transcript that was captured to the actual location of the tissue that was lying on top of this specific capture area here. So if we look at these barcoded spots in a little bit more detail, each of these spots has a diameter of 55 micrometers. and a distance center to center of 100 micrometers. And the 55 micrometer diameter gives you the an idea of the resolution of the system. So depending on the tissue that you're working with, you'll capture anything from 5 to 15, 20 cells within this one barcoded spot. The oligos that are hybridized on the glass slides within the barcoded spots contain a partial read one, the spatial barcode really labeling the origin of the transcript, a UMI, unique molecular identifier, and then the poly-DT tail, capturing the polyadenylated RNA, either directly or in case of the probes via this artificially or synthetic poly-A tail that we introduce with the probe pool. When we look at the workflow, you really don't need to read every single step specifically and what's entitled. It can be completed in 1 day or 2 days for the FFPE workflow, plus sequencing at the end and analysis. But with this slide, what I wanted to highlight is that really between the manual and the CytAssist workflow, there is very many similarities. The only step where the CytAssist comes into play is the permeabilization and probe release step, which is handled or taken care of inside the CytAssist instrument. Outside or prior to the instrument, for FFPE tissue, you deparaffinize immunofluorescent or H&E stain your section and take an image. And that is followed by decross-linking and the probe hybridization step overnight on a specific PCR machine. So very familiar steps following that. On the slide, probe extension, then you transfer to the tube and perform a few additional steps in the library construction part, and then you're ready to go and hand it over to sequencing. And just a couple of examples here to showcase what glioblastoma sample, for example, looks like on our new Visium gene expression slides with 2 capture areas. And we can see here on the small area in terms of the size that we can cover and the clusters that we can identify. And then we've used the same sample, but utilize the larger capture area, Visium slide, 11 millimeters by 11 millimeters. And, you're able to actually identify more cells and identify more clusters here in this specific example. And for the glioblastoma section here, looking or focusing now specifically on the 11 millimeter slide, we've looked at a large mess, or we can see a large mess of proliferating cells, which is positive for cell cycle markers we've used. And we do see this mess has an upregulation of oncogenes cMyc and mMyc. And it's interesting to note a small region has also high level of cMyc, and this region is starting to gain Ki67 signal, suggesting that this could possibly be a new form proliferating foci in the tissue section. And using Ward's clustering, we see the new mess has similar gene expression signature as to a small population of cells in the larger proliferating mass. So just a few examples for Visium, and just summarizing what Visium spatial gene expression allows you to do on top of transcriptomic analysis with morphology together. You get really high resolution, even if it is not single cell resolution, but you do get on average 10 cells per spot, depending on your tissue size or what on your tissue and what is covered in the 55 diameter area. And we see a lot of researchers combining single cell data with Visium spatial data to really increase the resolution further and to really enable the identification of cells that are captured in each of these spots. With the new Visium Spatial FFPE option, we do see that there is no tissue detachment happening anymore. And obviously you don't need to dissociate your tissue for spatial gene expression. And detection of protein and transcriptome is enabled currently with immunofluorescence, and it's going to be enabled with larger antibody panels in the near future on the CytAssist instrument as well. And we think it's important that we have streamlined data analysis pipelines available with example data, as I mentioned, example data sets, as I mentioned, and we'll also provide you some guidance on how to analyze single cell and spatial data. Again, bringing up the Space Ranger and Loop Browser capability for spatial analysis sets. You really are quite flexible with the way you analyze, because you capture the transcriptome across all of the covered barcoded spots in the software, and that's indicated here by the small little animation. You can select all of the spots that you're interested, or you just hone in on specific regions and keep reanalyzing your data set, you know, as you find it interesting and as you see fit. You don't need to pre-select any area of interest. And that brings me to the last short section, because I do want to highlight that specifically for a fellowship, but in general, we, the team from 10x Genomics as well as Millennium Science, here the science and technology advisors and field application scientists are here to really help you guide with your experimental setup, and also, you know, as you're doing your experiment. And we have a lot of support specifically for sample prep online for you to look at and read. You can dive into some of the webinars we've already run in the past, covering very specific protocols in more detail. You can read demonstrated protocols and tech notes online, and you can have also access to customer developed protocols and publications. And we have online technical support available if you have any questions coming up. We have talked about our analysis platforms. Just want to give you a quick overview that for each of the different assays that we have that I explained at high level to you today, we have analysis and visualization pipelines enabled, and for each of these we have further details on how they're done, how they work, again with sample data as well for you to familiarize yourself with. But then again, we are more than happy to talk you through some of these data analysis tools to just give you an idea of the different views that you can choose, where you can look at gene expression, you can look at the protein expression levels with your antibody selection, just custom group. You can look at experimental conditions. Really, there is a lot of options that Loop Browser initial analysis covers for you. The resources for informatics and software we've listed here. We have step-by-step analysis tutorials and guides, as well as webinars and documentation. And our bioinformatics experts are available to answer any of the questions you have specifically for a tissue or a sample type that you have, one analysis that you want to conduct. And this is really us. This is the team from 10x specifically. We also have Millennium Science field application specialists available with Paul Gooding. So between us, we'll really share guiding you through it. And Marta will now take over and explain the fellowship structure, the support structure, and how we really want you to apply for this. And we hope you do apply, and we really look forward to seeing some of your experimental suggestions and ideas that you have. And we really look forward to helping you meet your research goals. So with that, thank you very much for your attention, and I hand over to Marta now.
Thank you, Yvonne. I will now share my screen. Okay, and present. Do you see it well? Just asking.
Looks good, Marta.
Yeah.
Okay, thank you.
Thanks.
Just a quick note before I start. I have a very loud bird outside. I hope it will not interrupt this short talk. Welcome everyone to this short session about our Millennium Science Fellowship Program. My name is Marta Gabrielska, and I am a 10x Genomics account manager at Millennium Science, and also this fellowship program coordinator. So Millennium Science Fellowship Program, together with 10x Genomics, was initiated over 2 years ago, and we are very proud to be supporting more than 100 researchers over the years. And the program was born to address the need of creating a community of 10x Genomics users and give them tools and knowledge to confidently run 10x Genomics experiments, which especially at the beginning can be a bit daunting. So I would like to briefly say what are the benefits of actually becoming a Millennium Science Fellow. First of all, you can receive lots of training and mentoring session with both 10x Genomics and Millennium Science to prepare you for running experiments and ensure the success of these experiments to answer your research question. You also receive a personal care of the fellowship program coordinator, which is myself. And that means that any questions, queries, problems, you can send directly to myself. And if I don't know the answer to the questions, I will find for you these answer people who will answer these questions. So this way you will never be left alone or feeling like you need help and the help is not provided. After completion of the fellowship program, you receive a 10x Genomics certified user qualification, which can be pretty useful for you to put in your CV and basically certifies that you are actually really qualified to run these experiments. If there is a need of, if your institute doesn't have the required instrument, there is a possibility of us providing a demo instrument, especially Chromium controller, possibly Visium Site Assist. However, it is subject to availability, so that about that we need to know early on during the fellowship so we can all plan ahead together. The part of the fellowship is also discounted 10x Genomics products, which is always good news when you start new technology. And every year we are running annual fellowship forum meeting, and you will receive a speaking invitation to that meeting. And the underlying need for creating the fellowship was to create a network and community of users. Therefore, by becoming a fellow, you join the membership, you receive the membership of the 10x Genomics Millennium Science Fellows Network and access to our exclusive information and networking Discord channel, which means that you have access to all the news, promotions, webinar information, and seminar information and support through that channel additionally. You will also have ability to learn who other fellows are and connect with them, find collaborators, etc. So if you are interested, what you need to do to apply? You will have to provide a project title and abstract, and there is no need to provide a detailed, lengthy description of your project. So I think it's one of the most straightforward fellowship applications. We are also asking you to provide a one-page CV and a letter of support from your supervisor. Also, during the application, you will be asked to fill a quick survey, which will allow us to learn a bit more about the project in context of using 10x Genomics technology, like number of samples, species, and type of assay you would like to run. There's some conditions to be considered when applying for this fellowship. So we encourage people with no previous personal experience with 10x genomics technology, because we want to provide the best support for new users who really need this training, mentoring, and guidance, as the existing users are already ahead of this step. Therefore, we are just encouraging people who never used it before. The fellowship and our activity is to support mainly graduate student, early to mid-career researchers, as we know it's really hard time in the research career. Therefore, we put it up to 15 years post-PhD, but of course, we are aware of certain exceptions. So if that's in your case, you can just provide additional information, and that will definitely be considered. This fellowship is also for scientists who will run experiments by themselves. Therefore, the whole fellowship is designed into teaching them and providing them guidance to run these experiments. And at the end, also, we will be asking you to commit to complete the training to ensure that you learned everything necessary to be successful in your experimental project. And therefore, we will provide a document to sign at the start of the fellowship. So what happens next? If you're interested, please apply for the fellowship before the 23rd of December this year. When it happens, we will review these applications, and selected individuals will be contacted via email. If that happens, it will contain invitation to a nice and friendly interview with myself, where we can quickly chat about your project and your needs and just try to find the best solution and how to move forward. And official start of the fellowship will start with the onboarding meeting, where everyone will learn who is who to ask, what are the certain steps of the fellowship, and how we will move from there. And with that, I would like to say thank you. We know that not all those who wander are lost, especially in the science, because we are all from research fields. So by joining or applying for Millennium Science 10x Genomics Fellowship, you are joining a really enthusiastic group of researchers, and we are there with you, basically holding your hand during your scientific journey. Thank you for that. And now I will stop sharing, and we are happy with Yvonne to take any questions if there are any.
At the moment, we don't have any questions, but we have a raised hand. So I've just unmuted you. You can unmute now and talk if you want.
Hi.
Hello.
Hi, guys. Thank you very
Hi.
much for the lovely presentation and the introduction to the fellowship. And my question is about the success rate of the application for fellowship. Thank you very much.
Basically, the applicants whose projects look feasible and like really suited for 10x genomics technology have very high chance of being accepted to the program. So yeah, that's the rate is really high. We don't reject applicants if the project is great. We are basically really happy to support you.
Thank you so much.
Thanks for joining, everyone.
Thank you.
And hope you all apply. Good luck.
Bye.